So I think I'm online again. At least I hope that everything works out again. Hello and welcome again. Microbehunter here. My name is Oliver and welcome to the weekly Microscopy live stream, Saturday live stream. Again, as always, please post a comment if you experience any sound problems or technical problems. And today I would like to show you a few sources of so-called safe microorganisms. What does this mean when I say safe microorganisms? Well, there are many people who would like to put microorganisms under the microscope. Maybe they're hobby microscopists, or maybe they want to do microscopy in a school or with children. And in this case, we would like to look at bacteria and fungi, but the sources should be safe. And what do I mean with when I say a safe source? Of course, it's possible to take some food and then wait until it starts to rot. And then you have, of course, got fungi growing on the food and bacteria growing on the food, and maybe it starts to smell a little bit bad. And then when you take some of this rotting food, this spoiled food, and put it under the microscope, of course you're going to see then in many cases many fungi and bacteria and so on. But it's a little bit it's a little bit disgusting sometimes, maybe, and you don't really know what these fungi and bacteria are. That's actually a problem, because sometimes they make spores, and when you inhale those spores, then some people might get certain reactions, and that's not so good. So for today, what I decided to do today is I decided to put some food that basically have fungi and bacteria growing on them under the microscope, because those fungi and bacteria can be eaten. Okay, so that's basically biotechnological products, and some of them indeed are an important part of the food, and of course they are safe. And so what I brought along today is a couple of samples and specimens. I would like to show you what they are. Of course, depending a little bit from what cultural background and from what country you are, you might already be familiar. But these are maybe some foods you might not be so familiar with. Well, this here, for example, this white thing here, that is yogurt. Okay, I think this can be found in many countries. And yogurt is, of course, made of milk, but they added bacteria. And bacteria, the bacteria that were added to the milk, they start to ferment the milk. They break down the milk sugar, they form an acid, lactic acid, and then basically you have, after a few hours, you have yogurt. And this yogurt here is full of yogurt bacteria. And there are different types of yogurt bacteria, also depending on the type of yogurt that you're interested, that you want to make. And that's basically billions and billions and billions of bacteria are now in this yogurt over here. And of course it's safe because of course we can eat it. It's not a problem. And over here so I'll be putting a little bit of yogurt under the microscope a little bit later. So let's put it away for right now. And over here, I've brought along, here, this here is cheese. This is called Camembert cheese. Again, maybe some of you already know it, but depending on from what country you are from, you might not be familiar with this. It's one of my favorite cheeses, and it's also made of milk, of course, but what you can see is that there's this white, fluffy coating material on the outside, and that is a fungus. So that's a mold, and what happens is that the mold grows on the surface, and the mold, the fungus, produces enzymes, and these enzymes are given off into the cheese, and essentially the cheese is being digested by the fungus which grows on the surface. And this gives the cheese its characteristic taste and smell. And what I'm going to do later is I'm going to take a little bit of this white fluffy fungus and put it under the microscope, and then we can see the cells of the fungus. Okay, again, very important is the following: is that the fungus that's growing on the cheese, of course, was added by the company. So this is not just some kind of a wild fungus that happens to just fall on the cheese, but actually it has been added and it started to grow there. But if you now see other types of mold or other types of fungi growing on the cheese because it has different spots of a different color, then you have to throw the cheese away. Yeah. Now, you might wonder, why is it possible that some fungi can be eaten and others not? Well, and this depends whether the fungi are producing certain poisons. Of course, the fungus that is growing on the cheese here does not produce any poisons, and therefore we can eat it. Okay, so I know that some people actually call cheese like spoiled milk, and strictly speaking, yeah, that's not so wrong. Yeah. So another type of cheese is blue cheese, and you can see that there are blue-green spots in the cheese, and that's also mold. So it has also been added, of course, by the company, and you see that here, in this case, the fungus, the mold is growing now inside the cheese, and here it's growing on the outside, but the principle is the same, that the fungus produces enzymes that are given off they're given off into the cheese, and this basically gives the cheese its characteristic smell and taste. This one, for example, I'll take it. It smells extremely strong. Okay, so yeah, it's a very strong, has a very strong smell. Yeah, so what I'll be doing is using some toothpicks. I'll try to extract some of the fungal cells from the blue-green spots over here. Okay, so this and over here, this is a classic. Okay. This here is yeast. It happens to be the dry form, dry yeast, and I can show you. Yeah, and it is in powder. Yeah, so there is. Yeah. Okay, where is it? Yeah, here it is. Okay. Yeah, yeah, it has a skin color. You might not be able to see it quite well, but yeah, here now a little bit. Yeah, and this is dry yeast, and yeast is also a fungus. But it looks a little bit different because the fungus growing on the Camembert cheese has cells that are very long, and also the fungus that's growing here on the blue cheese has very long and filamentous cells, while the cells of the yeast are rather round. Yeah? So, but it's again a classic, and this is a very easy to observe specimen because, of course, it's just pure fungus. Because when I take off something from the blue cheese, sometimes I'm also taking along a little bit of the cheese itself. And this kind of makes it difficult to distinguish the fungus from the rest of the cheese. Yeah. So, and last but not least, I wanted to show you something else today. Something that I also recommend. Look at this here. This is something that I bought as well. I bought it from Amazon. And these are also yogurt bacteria, but they're in a freeze-dried form. So it's a powder. Yeah, and what you have to do is you take a very small amount of this and you mix it with the milk, and then you put the milk on a warm place, and then the next day you've finished yogurt. Yeah, so these are also yogurt bacteria, but they are in a freeze-dried form. It also, just like the yeast, it has the advantage that these are pure yogurt bacteria. Yeah, because when I put the actual yogurt under the microscope, yeah, then I will also see a lot of milk, milk proteins and milk and so on, which kind of might make it sometimes a little bit difficult to distinguish the bacteria from the rest of the yogurt. But if you have it in a pure form, then of course it's much easier. Okay, yeah, I've already received a few comments, mostly welcome messages. Hello, hi, hello. I'm getting hungry again. Yes, cheese. And someone's excited for the viewing. Yes, I also like doing live streams a lot. And you know what? I don't know. Let's just start off with the yeast, because it's just happened to already put it into the container, and if you into the beaker. If you want to observe yeast, then of course you have the possibility to use dry yeast, just like this one over here, or fresh yeast. And what you have to do is, if you use dry yeast, of course you add a little bit of water. Okay, you can see that the yeast immediately starts to dissolve, but I'm going to give it a few minutes. Okay. a little bit for the yeast to dissolve. And something else that you might want to try is to add a little bit of sugar, because if you do that, then the yeast will start to break down the sugar to carbon dioxide, and you have actually CO2 bubbles forming, and you can actually also observe this under the microscope. Yeah, another comment here. I wish I had a biology teacher like you at school. Haha. Okay. I'm going to comment on that as well. One of the reasons why I am doing those live streams is because some of these things is kind of difficult to do in school. Because in school you're always pressed for time. You have a syllabus to complete. Therefore live streams and YouTube allows me to share my passion for biology in a different way. And when you are in school as a teacher, then you have to communicate facts, and then you're standing in front of the chalkboard, and then you have to draw diagrams, and then you have to prepare the students for tests and exams. And of course, then sometimes I feel that the actual biology, the hands-on biology rather, sometimes falls a little bit short. Live cultures, these are there are a couple of is it normal blue cheese? Yeah, this is a pretty normal blue cheese. I'm reading off the comments right now. It's again, it's just a local brand, and they're different types, but they all kind of look the same here. And kombucha, yes. As a matter of fact, kombucha and kefir. I don't have a kombucha and kefir with me right now, but that's also a very good and interesting thing to look at under the microscope. And this is not Roquefort. Roquefort cheese is also possible, but it is just called, they call it, I don't know, it is just a regular blue cheese. Blue moldy cheese. But I would say you can be a little bit creative about this and try out different cheeses. And I'm going to simply do now the following. I'm going to take a small sample of the yeast. And I would say that this should be or could be one of the first specimens to observe if you have a microscope, because it's a little bit like a benchmark. Are you able to see those cells or not? So yeast cells are eukaryotes. So this means that they have a nucleus. They're fairly large. And when you prepare a slide like this, still be aware that don't use too much water. Otherwise, the cells keep on floating up and down, in and out of focus, makes it kind of difficult to observe. Okay, so I'm going to go on here. Homeschool my children, and we often follow and reproduce the experiments. Thank you. That's also very nice feedback. Yes, have all the blue cheese the same species of fungus? I probably would say, without that I know it, I probably would say no. And it could even be that sometimes there's a combination of different species on the cheese. So as a matter of fact, I know of a publication I once read many years ago, where actually scientists also tried to isolate the different types of bacterial species growing on a cheese, especially maybe if the cheese ripened naturally. And if they did not add any cultures specifically, then you can actually find out that sometimes the cheeses are a very complex mixture of different types of bacteria and fungi growing on it. But in this case, what I've got over here, of course, they added those fungi to it. Okay, which magnification do you recommend for fungi? Honestly, I would say 400 times magnification or 600 times magnification is the maximum that you need. Depends on the fungus. You were going to see that the Camembert cheese, actually the cells are pretty large, and for the blue cheese, it's a little bit more difficult. Okay, so and I looked at recently bought kefir under the microscope, but all the bacteria were dead. Just because they do not move does not mean that they're dead. Okay, not all bacteria move. Okay, so movement is not movement or lack of movement is not a sign of whether the bacteria are alive or dead. Okay, so this is actually one of the problems of when using microscopes, because sometimes you just don't know because if they're alive or dead. So let's do the following. I have to change, of course, over to the microscope view. Okay, so that you can always see the magnification here. This one is now a 4 times, 10 times, 20 times, and then you're able to see how this looks like. And that's basically. Let me put the cheese away here. Up. we're going to put this somewhere over there to the side. Okay, so yeah, so that's basically yeah, just a mess, right? Because the magnification is so low, you see a couple of clusters and clumps here. So let's go up. Now it's 10 times magnification, and I have to refocus a little bit. Okay. And I think that honestly, I can already see that there is this movement going on. This is because of the flow of the liquid. But honestly, this is not a very satisfying image yet, because I think that the density is still a little bit too high, and I'm not able to see the individual cells. Okay, so let's go up with the magnification yet further, and here we start. It's starting to become a little bit more interesting. You might be able to see those round little structures. Now these are the individual yeast cells. Okay, so this is now what is this, 200th times. I mean, with the magnification, one has to be a little bit careful, because it's the camera that I have up there which actually captures the image, and it's a 20 times objective. And let's go up to 40 times. And here, this is where you can now start to see the individual yeast cells. I'm going to open the condenser a little bit. Okay, and I'm going to go yet further up. Yeah, and now we're able to see, of course. Let me change the condenser setting a little bit. And I have a DIC microscope for differential interference contrast, which of course also increases the contrast a little bit. So, and this is now all of those oval, round, egg-shaped structures here. These are the individual yeast cells, and what I recommend, if you try to replicate this experiment at home, what you do is try to add a little bit of sugar, wait a few hours, and then look at them again, and you might see that some of the yeast cells start to divide. And this is called, what they divide in an asymmetric way. It's called budding. So you will then see one yeast cell, and then there is a small yeast cell growing out of it. It's called a bud. And it looks like it kind of comes off of the parent cell, but genetically they're the same. So this is a form of an asymmetric cell division that yeast does, and this is called, this type of cell division, mitosis, is called budding. But we do not see it here, because the yeast was just dry, in a dry form, and now takes a little bit of time to become active again. So I'm always looking over to the comments again. Okay. I want to see that cheese bacteria. Good evening from a ship in Sweden. I love it. It's so from all people from all over the world. Recently found your channel, and it's really getting me into microscopy. I'm into amateur astronomy, and it's interesting to see so much overlap with microscopy. I have to be honest with you. Many years ago, I was also very active in amateur astronomy. Bought myself a small telescope, watched the stars, watched the planets of Jupiter, and of course the moon. And that's of course it's enthusiasm with science, I think, that we all of us share. So you can see that over here there are some blurry patches, and when I refocus, you can see that there are still some clusters of yeast cells that have not yet dissolved. And I should have actually done the following. I'm not going to do this now, but if I should have added a little bit of sugar. And when you do that, you might actually see bubbles of carbon dioxide, because what the yeast does is it takes the sugar and it will break down the sugar and form CO2, carbon dioxide, and therefore you will actually see CO2 bubbles, and they will actually grow. And this is something that you can actually also see quite well under the microscope. The objectives on your Olympus appear to be high tech. Well, they're pretty decent. If some of you might be interested, if you're a little bit more interested in the technology of the microscope here, this is my 60 times microscope, and if you look very carefully, it has this ring here. You see this, and I'm able to turn this ring. Okay. And you might be able to see also that the clarity and the sharpness changes a little bit. And what this ring does, it adjusts for the correct thickness of the cover glass. Okay, so the numbers that you see down here is actually the thickness of the cover glass, so 0.20, 0.23. Because this is a high resolution, high aperture, high numerical aperture objective, and they're very, very sensitive if the cover glass does not have the correct thickness. And here you're able to adjust for the correct cover glass thickness, and you can see that, yeah, in some, yeah, the clarity changes a little bit, okay? Yeah, and then you have to find the best position, and I think this gives the best result here, okay? Yeah, so is DIC better than phase contrast? You cannot, well, okay, explanation here. DIC stands for differential interference contrast, and this is the following. If you look now very carefully at those yeast cells, you're going to see that on the top corner, it looks like they are a little bit darker than on the other side. So it looks like that the light is coming from one direction. This is kind of how it appears. This is actually not quite correct, that it has this three-dimensional, it gives an appearance of a three-dimensional appearance, but this is not because the shape of the cells really is like that, I have to say, okay? But this allows you to increase the contrast, and this is, I have also up here on the microscope, which you cannot see, I'm able to shift a prism, and now look what happens when I change it around, and you can now, yeah, make it appear as if the light is now coming from the other side. Now the bottom right side of the yeast cells, they have a shadow, and the top left side is bright, and when I turn it around, it looks like that the light is coming from a different direction, okay? But that's because I'm shifting a prism, and this increases the contrast. But the question is what about phase contrast? So thank you for asking. I'm going to show you now phase contrast. But phase contrast I only have with 40 times. So this is now 40 times, okay? DIC still. And now let's have a look at phase contrast at the same magnification, okay? So I'm going to switch over. So this is 20 times, 10 times. Let me turn down the light a little bit. And here, this is now phase contrast. I have to change the filter. And I am going to open the condenser. I have to move out this here. And this is how phase contrast looks like. Yeah. So what you're able to see is that essentially phase contrast makes objects of a different refractive index and appear in a different brightness. Yeah. So this is basically how phase contrast looks like. Is it better or is it worse? You cannot say. It really depends on the type of specimen. I personally like DIC more because it retains more of the natural colors and it has a somewhat wider application spectrum. However, for certain applications, like if you want to observe bacteria specifically, probably phase contrast is better than DIC. Did I misspeak now? DIC has more general purpose applications, I think, okay? So, yeah, so add sugar, I want to see it. Oh my gosh, you know what I'm gonna do? I don't have sugar with me, I totally forgot about this. I'd have to go over and run into the kitchen. So I'll do this a little bit later if I have time, okay? Some people with aquariums use a do-it-yourself carbon dioxide system using yeast sugar to create carbon dioxide to dissolve it in water. And I have done this myself. When I still had a home aquarium about 20 years ago, my plants didn't want to grow properly. Because there was too little carbon dioxide gas in my aquarium. So what I did is I've taken a 5-liter container of water, added sugar and a little bit of yeast, and then for about 6 weeks made very, very tiny CO2 bubbles, which I've fed into the aquarium to actually make the plants grow better. Yeah. So this is again now DIC. I have to switch over everything again. All of the settings I have to modify again. Yeah. And here we are again 40 times. With DIC, you have to refocus again here a little bit, okay? Okay, so you can have DIC and phase contrast on the same microscope. Yeah, the better yes, of course, the better microscopes are completely modular and you can configure them based on your needs. And I happen to have an extra space here, so I contacted the company and told them, please send me a phase contrast, 40 times phase contrast objective, and that's what I got here. Okay, so let's move on a little bit. Okay, so this is yeast, and yeah, here this is again the 60 times that we had a look at, and let's move on. Okay. So what time is it? I always have to check the time. Oh, we've already talked 20 minutes. So yeah, I'd like to now do the following. I'd like to put some of the yogurt now under the microscope. And of course, there are different types of yogurt, and I've chosen this one here because it is extremely solid. Okay, some people like more solid yogurt, other people like more creamy yogurt. But this solid yogurt does have a really nice advantage. See how the liquid separates so nicely. Okay, so what I want to do is I want to put some of this liquid here under the microscope. Of course, I can also take a small sample of the yogurt directly. Okay, we'll try this as well. But I think that some of the bacteria, yogurt bacteria, might be covered up by the yogurt itself. So I think you maybe are going to get a better result if you take the liquid. Okay? Yep. Can microscopy be used to confirm a streptococcal throat infection from a sputum sample? Well, I would say the following. I mean, of course, if you take a sample from the throat and you see a lot of streptococci from the shape, yes, but generally I would probably say that for diagnosis, you'd need more tests just to make sure. Okay, so what I'm going to do now is I'm going to take some of this yogurt, here as it is, and next to it, there's still a little bit of yogurt in here, but let's try just a little bit of the liquid. Okay. Let's see if there is a difference. And the cover glass goes on top. And I want to just tell you that this here is bacteria difficult. Okay, bacteria are difficult because they are small. They're pretty transparent, and you need a reasonably decent microscope. When I say that, you need a microscope at least with a condenser, and the magnifications also have to be a bit higher. Another thing that I just want to say is the following. These cover glasses, you might not be able to see them because they're are a little bit dirty. Okay, they are new, but sometimes during the manufacturing process, for whatever reason, they are a little bit dirty sometimes, and especially when I look at bacteria, because they're kind of difficult to observe. I sometimes consider it good if you dry wipe it, simply to take a little bit of the grease off in case you touched it on some of the dust and dirt, and you're actually going to see that it actually becomes much more transparent. So I'm going to do the following. Look, I'm just going to drop this one on here. The other one I'm also going to dry wipe, and I've already used too much yogurt here. I can already see that. Here is okay. Look how the liquid immediately spreads, and here what I have to do is I gotta press this. Okay, and of course there's gonna be yogurt spilling out on the side, and that's okay, but I want to remove it a little bit because I do not want to dip my objectives into the yogurt, and I'd have to go up anyway a little bit with the magnification here, so the distance between the cover glass and the objective is not very high, so I just want to play it safe. Okay, so let's use the back here, and I think this should be okay. Okay, let's wipe the liquid away, all of the excess liquid. Okay, so and let's give it a try. Okay, but I just want to also tell you a little bit that it's not going to be quite as spectacular as the yeast because the cells are quite small. But let's have a look anyway. Okay, so I'm gonna switch over again to the microscope view. Let's remove the the yeast. Okay, let's turn down the light a little bit, and here we go with let's do the left one first, the yogurt itself. Okay, and I'm just gonna as expected, need to refocus. Lots of way too dense. Okay, so let's go to the side here, that's better. Okay, you see that there are these clusters, that's the actual yogurt, and then those white areas in between, that's what I actually want to look at. Okay, so I'm gonna go up now to 20 times. Okay, and 40 times. Let's see if we are lucky here a little bit. Okay, I'm looking at this gray area here, and I'm going to and I know you're going to be hugely disappointed, but those little dots that you see, these are bacteria. Okay, actually there are not that many. I've actually had just before. Here it's better. And if you look very here, it's better. Yes, here we go. Okay, you see that some of them are longer. These are so-called rods. Yeah? These are all yogurt bacteria. Yeah? And they don't move because they don't move. This does not mean that they're dead. As a matter of fact, they are alive. If I were to take some of the yogurt and mix it with milk, then of course I'm also able to make new yogurt. And what I'm going to do now is I'm going to go up to the 60 times. Okay. And I need to change the filter setting again. And again, that is DIC. If you look very carefully, then you might be able to see that some of the bacteria are actually round and also in chains. Yeah? So, yeah. So this is not phase contrast, okay, but DIC. Yeah? Very typical shape of bacteria here. But it's very, you see, the contrast is kind of low, okay? So, yeah. So let me try the following. I'm going to close the condenser a little bit to increase the contrast. And I have to always go up with the light as well. Ah! Do you see this on the bottom here? Looks like a chain of round bacteria. Okay, of cocci. So maybe I can actually move this a little bit more into the center. Yeah. Because if you agitate, if you mix the yogurt, then you will end up breaking those chains, okay? So, and for this reason, yeah, you got to be a little bit careful, but when you take it out carefully, then, and here also at the top, my finger again is, yeah, sometimes you see that they're double, yeah? But it's these are, and you see that because there are different shapes here, there are different different bacterial species. Yeah? So I'm going to go over again to the comments over here. Can microscopy be used to confirm this? Oh yeah, I talked about this. If you can identify the bacteria, yes. The point is the following: the shape of the bacteria alone is rarely enough for you to identify them. Yeah? You cannot, the analogy that I always use is you cannot determine the make of a car or the brand of a car by looking at its color. Yeah. So, and it's a little bit the same also with bacteria. You cannot identify bacteria. Look here, this one over here is two cells next to each other. Two, yeah. No, not disappearing over my shoulder. Yeah. Yep, if you can find the time, I would very much like to see you show the difference between algae and cyanobacteria from the sea. We always have a huge problem in the Baltic Sea every summer. Yeah, just for your information, thank you for the question. Just recently in my other channel, I did make a video about cyanobacteria. And sometimes, of course, it might be difficult, but generally, algae, real true algae, they have cell organelles. So this means you should be able to see chloroplasts. Yeah? But then again, yeah, the biodiversity is quite large. Yeah. Rods, yes, long bacteria that you see are rods, okay, and the round ones are called cocci. Do you ever buy or make Petri plates for getting isolated cultures? Short answer, no, not anymore. When I was still at university working on my thesis, I've made a lot of Petri dishes. However, I'm not doing this anymore. And I also discourage this for the reason that, strictly speaking, depends a little bit on the country that you live in. If you want to grow bacteria on Petri dishes, you have to be an authorized lab. Because if you do not grow bacteria that you do not know what they are, strictly speaking, you are already in safety category 2. Safety biohazard category 1 is safe. So if you, for example, work with known safe bacteria, and category 2 is already unknown bacteria, and there are a total of 4 categories. So I do not see a need to grow bacteria myself now. I know that some people are doing that at home. Yeah, I probably wouldn't. Yeah. Are the chain bacteria did not separate after cell division? Yeah, so that's the whole thing. Some the cells, you've got a coccus, okay? A cocci around bacterium and divide, and sometimes they stay connected together, okay? And sometimes they will stay together as a so-called diplococci, 2 of them, and then they will separate. Some of them try to stay in chains, and what I also found out is that sometimes, yeah, they stay in chains, but when you mix the yogurt, they will break apart. Yeah, so it's not always very stable. Look at the top left corner. There is also a chain. I cannot point towards top left corner. I'm going to move it now into the center. Yeah. It's still now in the top center up. Yeah, now it's pretty much in the center here. Let me see if I can point towards it. Yeah, at my fingertip here. Okay. It looks like a long cell, but if you look very carefully, you're actually going to see that these are several cells. Okay. It seems to be like a small chain. I hope at least I'm able to see it like that. I don't know if over the YouTube compression whether you're also able to see this resolution. Yeah. Yeah. Bio level 2 when you have unknown bacteria in a petri dish. Yeah, at least this was the law of that when I was working in the lab, I was isolating bacteria. So it's part of my thesis from air, and we had to identify them. So it was an air quality assessment, and we had frequent inspections of whether we are actually working according to the rules, because we were working in an elevated biohazard category because we didn't know what we're working with. Yeah. Which evolved first, bacteria or yeast? If yeast came later, did it evolve from bacteria? This is for this question there is an answer. Very clear. Prokaryotes, bacteria came first. And there is basically the cell organelles like mitochondria and chloroplasts in algae. These once were separate bacteria that moved into the cell. This is a concept called endosymbiosis. So the mitochondria that we have, these are the powerhouses of the cell, once were so-called proteobacteria, purple bacteria, and as a matter of fact, chloroplasts and the mitochondria, they have their own DNA even. Yeah, so so eukaryotic cells like plant and animal cells, they came after bacteria. This is actually quite well documented. There's genetic evidence for this and a variety of other scientific evidence. Yeah, the concept is called endosymbiosis. Okay, how do they look like under bright field and dark field illumination? Unfortunately, I cannot show you dark field, but thank you for the bright field comment. All I have to do for bright field is to pull out the little condenser polarization, and I'm gonna go down with the light intensity. I'm closing the condenser right now. Maybe this was too much. And this is now bright field, and what you're able to see now, so basically most of you should be able to see it like this, okay? But what you're able to see is that the bacteria, they seem to have some kind of you're only able to see the border of the bacteria, right? And this is because of the difference in refractive index. So this is actually, these are so-called diffraction patterns. Yeah, and actually, strictly speaking, these are so-called artifacts because bacteria don't have this type of border around them, but it actually helps you to see them. Now look what happens if when I open the condenser, okay? When I open the condenser, of course it's becoming brighter, so I have to go down with the light, yeah? But you see also that the contrast goes down, yeah? Because the diffraction patterns are less. Yeah? So if you see, it makes it appear not quite easily visible. Yeah? And for this reason, techniques like phase contrast and so on have an advantage. But what I'm going to do is I'm going to just switch over to phase contrast now, okay? So this means I have to change this 40 times, 20 times, 10 times, 4 times, 40 times phase contrast, okay? And I have to change also over here the filter settings until I have the phase contrast annulus in place. And look, it's too dark. I have to go up with the light intensity and refocus. And what you're able to see now, and that is basically why people who want to observe bacteria, why they like phase contrast so much, is because now I'm seeing the transparent bacteria. I'm seeing the transparent bacteria darker. Okay, so you see all of those lines here, all of the rods, yeah, are now much more easily visible. Yeah, and there is also less diffraction. Yeah, you might also, if you look very carefully, see that the bacteria, even though they are darker, they are a little bit bright around them. Yeah, and that's also very typical of phase contrast. Yeah. So yeah, so this is a 40 times phase contrast, and I would say that phase contrast is, yeah, you see, it looks more black and white, right? And that's the whole point, because phase contrast is able to change differences in refractive index into brightness differences. Yeah, and the physicist, yeah, what was his name? Fritz Zernike, back in the days, he invented the phase contrast technique, and for that he got the Nobel Prize. Yeah, yeah. So that is yeah. So what makes some bacteria highly deadly? Okay. It's also not only bacteria, also some fungi. Very briefly, because some bacteria produce poisons that actually cause a problem. So I'll give you an example: the tetanus. Tetanus, back in the day, people had somewhere a wound and it got infected, bacteria got in, and this happened to be Clostridium tetani. That's the name of the bacterium, and those bacteria produced poisons that spread and caused the people to become paralyzed. They had this called spastic paralysis, where the muscles all of a sudden started to contract. Yeah, so this is yeah, indeed the muscles contracted involuntarily, and the people of course then died because they also couldn't breathe and so on. And this was caused by the tetanus toxin. Yeah. So the answer, what makes some bacteria dangerous or deadly, same with fungi, is because some of them produce poisons, and depending on the type of bacterium or fungus that you eat, yeah, this can result in different problems. Yeah, I use your trick to change my microscope with dark field, and it cost me nothing. I was not able to get it to work with 4000. Yeah, of course, and I can tell you why. It's not powerful enough. So it's my microscope here only has dark field for the 40 times objective, so this is not going to work quite well. For the 4 times objective, sorry. Dark field is when you include a little filter, and then you see the bacteria or whatever you look at bright on a dark background. And this basically allows you to see also very structures that you might otherwise not see. And it's very cheap. And the reason why dark field is very difficult with higher magnifications, like for example with a 40 times objective, is because it takes away so much light. And you also need to make the central disk of the dark field filter larger. Yeah? So at high magnification you already have little light, and then you're making the disk which blocks the main light even larger. So yeah, you might want to try it out and then use some kind of long-term time exposure using the camera. That would probably work. Okay. I actually wanted to show. Look at the other. Look, people. I'm still with the first Yorid sample over here, but see there's the other one on the other side here. The one with the liquid only. Yeah. Yeah. I see mostly over here. Yeah, let's see if I'm able to find bacteria here as well. This is where there are mostly only the liquid. So I'm going to close this again. I'm going to do it like this. I have to increase the light. Why do I not see anything? Because I've got the wrong filter. There are multiple things that I have to adjust here, you know. And it's kind of dark because I need to increase the light a little bit here. Oh, okay. Yeah. This is same story. Yeah, but less of the yogurt itself, now more of the bacteria. I think you get the idea. I think we can slowly move on. I'm interested in the matrix of bacteria and yeasts formed in kefir. Would it be considered a colony? Is it a combined endeavor to form the matrix? Yes, kefir. I did some research on my other channel many months ago. I did actually put some kefir under the microscope. Is actually a complex aggregate, symbiotic relationship between fungi and bacteria. Yeah. Is it a colony? Well, it's a little bit a question of definition. In a traditional sense, you refer to a colony if it's pure bacteria growing on a petri dish. Yeah, I would say it's what I read is it's in a matrix of polysaccharide which holds it together, and it's a complex mixture of fungi and different bacteria, kefir. Yeah. Are you using all APO, apochromatic objectives? That is the question here. Are you using all APO objectives? APO objectives are apochromatic objectives, and those objectives are basically give you the best image quality, and the short answer is no. I'm not using apochromatic objectives. I'm using semi-apochromatic objectives, also known as fluoride objectives. They are also pretty good, and they basically, apochromatic objectives would have been too expensive, so I didn't even, I didn't bother. And but these are semi-apos, so-called fluoride objectives, and they're also fine. So let's move on. So this was yogurt. This had some yeast before. If you just joined in, because I've seen that some folks have joined in just now. What I'm doing today is I'm putting some cheese and yogurt and these things under the microscope, because these are all foods, and all these foods contain microorganisms, bacteria and fungi, and because they are part of the food, they're safe. Because I don't want to spoil food and put that under the microscope. I'm not saying that's automatically always dangerous or so, but generally, especially if you want to do microscopy with children, or for homeschooling, or for hobby use, or if you just want to explore a little bit the food that you eat, then why not simply use those types of food that already have microorganisms growing on them. So I've got two more things that I'd like to put under the microscope. Two cheeses, and of course this one. You know what? Let me put this on here first, okay? The reason being because these are also yogurt bacteria, but they're freeze dried. And because I was just looking at yogurt, why not stay with yogurt? And I'm just gonna show you how I'm doing that. I need to take a slide, okay? And usually what you need is you need a small amount of water, and I don't have a pipette now. Okay, here it is. Here's some water. So here's my pipette. So I'm going to put a little bit of a drop of water here on the slide, and I take some toothpicks. Where are they? Here, toothpicks. And I will simply take some of those freeze-dried yogurt bacteria. They sell them on Amazon and all over the place, but actually it's not necessary. If you want to make your own yogurt, just take a spoon of yogurt and mix it in with some milk which you boiled before. But am I able to get something out here? What's going on here? Okay, you know what, I think there must be already enough bacteria small, so I guess, let me have a look. Yeah, I can see it's a little bit cloudy already. You don't want to have too many of them anyway, because otherwise there are so many of them that they end up covering each other up. So I put now some freeze-dried yogurt bacteria, from the yogurt culture on here. I'm just going to trash this here. Cover glass, very important. Again, when you want to observe bacteria, you really use only very little water. The reason being, otherwise they float up and down, and then you lose focus. So really, very little water. Probably is already too much anyway. Yeah, but maybe it's gonna work. Okay. Yeah. So let's have a look at this again. Switch over to the microscope view, and let's have a look here. Okay. And okay, see all of those. This is all dirt. It's all dirt from the slide, and now I have to kind of see. I don't know what this is. Yeah, these are droplets of dirt in there. Yeah, here. So okay, this might be now. Let's have a look. And not all of them have dissolved, I think. But those little dots that you see. Okay, again, up with the magnification a little bit. I think the other thing worked better. Yeah, here. These seem to be okay. Here we go. Okay, all of those dots that you see. But those bacteria, again, they need some time now to become active because they're freeze-dried. Yeah. So let's go over here again to 60 times. Change the filter setting a little bit. and maybe open it a little bit here again. Yeah. I think the others of the fresh yogurt, they looked a little bit better because they do now take a little bit of time to actually start to regenerate. So, a few more questions that I would like to answer here. If you add yogurt to a Petri dish with bacteria food, can you then control the culture? I tried doing this. Actually, I tried growing yogurt bacteria on a Petri dish containing yeast extract, and it didn't work because this was not the type of food that the yogurt bacteria want to eat. Probably have to add some lactic acid, so you have to make sure that the agar plate has the correct nutrients, otherwise they don't want to grow. Yeah, and of course yeast extract is not, they're optimized to grow in milk, and yeast extract alone was not the right, so I did not get any decent colonies. Yeah, I tried this already. Yeah, what came first, the yogurt or the yogurt? Yeah, the question is a little bit this chicken and egg problem. You see the following. When you allow milk to be spoiled, just let some milk stand out in the warm without adding any yogurt bacteria, you're going to see the milk spoils, and in some cases it will turn solid just like yogurt, because the natural bacteria in the milk, yogurt bacteria, they will also start to multiply. So this means that they were already there all the time, but they have been now only cultured by humans for the food, specifically for food production. Also, kefir always existed, yeah, but the humans simply continued to culture them, and it kind of showed that they were not a problem. Yeah? Do you do a live stream every Saturday at the same time? Yeah, well, as of now, yes, but it could be that maybe in the future I might be busy on one or so, and then I'm going to tell you, but usually I want to do a live stream every Saturday. Do you see the stuff floating by now? There seems to be some kind of when the water evaporates, yeah, it pulls. Yeah. Have you used any of the PCO microscopes? No, I have not. Are they good solutions? Well, yeah, honestly, I don't know all of the brands. Yeah, and I would say that yeah, if the microscopes are used in education, in research, and so on, then not much can go wrong. Yeah, can yogurt by xylitol? I don't know about that. Okay. But that would be an interesting experiment. What you do is I don't know, you put some yogurt bacteria in milk and add some of the xylitol and then see if it yogurt forms faster or not. Yeah? A gram stain video. Oh, when I was still working in the lab, I did a lot of gram stains, but they're a pain in the neck. Gram stain, I have to tell you, because to get a proper result, at least one that is useful for publication, you have to get the bacteria at the right stage of the bacterial growth, otherwise they don't stain properly and so on. So it was kind of difficult. I had to repeat the gram stain over and over again until I got the results which were correct for publication. What is a gram stain? A gram stain is a traditional staining method to distinguish the so-called the gram-positive and the gram-negative bacteria. The gram-positive bacteria, they stain blue-black, and the gram-negative bacteria, they stain red. And this can or has been traditionally been used to kind of categorize the bacteria, because the gram-positive bacteria, they have there are certain bacteria that are gram-positive, for example Bacillus, and they have one cell membrane and a thick cell wall, and they stain black. And then there are other bacteria, so-called gram-negative bacteria, they have a very thin cell wall. So you can do, simply by staining the bacteria, you can do a rough, a very basic, I wouldn't say identification, but exclusion of certain bacteria. Yeah. So I would say that these days, if you really want to quickly identify bacteria, for example, in a medical situation, you don't do a gram stain anymore. You just sometimes they don't even test them, they just give you antibiotics, but there are other tests these days that I would say are more precise. Yeah. So gram stain has a strong historical importance, still is used for characterizing it, but yeah, I found it to be a little bit painstaking. Yeah. Yeah. Does staining kill them? Yes. Yes, definitely. Yeah. Definitely, you have to kill them, and you actually have to heat fix the slide, which means that you have to actually pull the slide over a hot gas flame to bake the bacteria to the glass slide, a little bit like a fried egg without oil is sticking to a frying pan. Yeah. A goat stomach was used for making kefir. Did it work because of just the organisms, or is the enzyme thing also play a role? Look, I think I'm going to hypothesize now. Of course, there are enzymes in a stomach. However, kefir you are growing separately and reproducing separately. So you have milk kefir, for example, and you take the kefir fungus and bacteria, you put it into milk, and it will ferment the milk. Okay, and you then isolate the kefir again, you keep on doing that. So there are no digestive enzymes of the goat left over, okay, because you're just working with a pure kefir. Yeah. So this is, how do you identify bacteria gene sequencing? Yes, as a matter of fact, this is one of the things that I have done when I was still working in the lab. If you're interested, actually I can do it's a little bit theoretical, but short answer. If you want to know with bacteria, you can do biochemical tests. Number one, so basically, what are the bacteria made of? The chemical composition. Or number two, a second way would be is what products, what food can the bacteria utilize, and what end products do they produce? For example, yogurt bacteria are able to use lactose, milk sugar, and produce lactic acid. You can exclude certain bacteria. And the third way that you can do that is you can do DNA analysis, gene sequencing. Okay, so that's a short answer. They still use gram stains all the time in clinical labs as a quick description of what you are looking at. Yep, because for example, certain gram-negative cocci might be some problem, right? So you can actually use this to kind of exclude certain things. But if you don't know at all what it is, and if you really want to identify it, then you have to take additional identification steps. So let's move on, because I still have 2 cheeses that I still want to put under the microscope. The hour is already over, but I'm not going to simply stop now, because I told myself that I want to show you this as well. So I'm just going to go on a little bit, maybe another 15 or 20 minutes. And yeah, this here is the blue cheese. Okay. Not so easy to put this under the microscope, because I have to pick up some of the mold and not so much of the cheese. Yeah, so this is a little bit of a thingy. So again, I'm going to put a drop of water on the microscope slide. I'm going to take next live stream, let's see DNA. Oh, I can do that. Thank you for the comment. I'll do some DNA extraction of myself. Seriously, I've actually already done this. Banana also works. Why not? But it's not. Yeah. Okay. Well, let's see if I'm able to also do a little bit of microscopy here. So what I'm going to do is I somehow have to put on my glasses first, because my eyesight is bad. And now I have to somehow take a small sample or specimen somewhere from this green-blue part here. Okay. You can see that the tip is a little bit greenish blue, and let's try to hope for the best. Okay, I don't know, maybe a little bit more. I'll use the other side here. I tried this before also, and I was not so happy with the result. Maybe this is gonna work better now, because I couldn't see a lot of cells really. But then last week I also tried this and it worked well. I tried it not in a live stream, but just for trial purposes. Okay, so here it is. A small drop of water, which you cannot see because the camera is not able to capture it. But yeah, over here again, I'm going to also quickly dry wipe it again, because I don't want to see, especially when, yeah, it's pretty dirty actually, especially when the specimen is difficult to observe. You want to make sure it's reasonably clean, otherwise the dirt and the oil and the grease and the grime kind of is a little bit distracting. And let's drop it on here, and let's again hope for the best. So where is my microscope view? Okay, so this is now 10 times. And okay, air bubble. Yeah. And what I'm looking for actually is so-called the hyphae. Hyphae are long, thin filamentous cells, which actually make up the fungus. And regular round structures, not the air bubble in the middle now, regular round structures which are actually the spores. So I'm looking for long cells, and here, which are basically part of the so-called the mycelium, the hyphae, and you can already see some of them over there, right? Some of the cells that are long. Because what a fungus is a fungus that has these long cells, and they kind of spread, making the fungus fuzzy. That's kind of the fuzzy part of and it kind of grows into the substrate, and as it grows, it breaks down the surrounding medium by releasing enzymes. And then when the conditions are right, it starts to make spores. So let's go up a little bit with the magnification now. Yeah, and again here, yes. So, and now I think, so forget about this large round air bubble, okay, but now this is now 40 times, and here we can already see that essentially there are these long cells here, these are the hyphae, and those regular round structures, these must be spores. Yeah? Do you think that the complexity of organisms in kefir has more health benefits than yogurt? Honestly, I don't know. We're eating so many, when we eat food, any type of food, we're eating cells, almost any type of food. So we're eating already very complex things. And it's all digested anyway. So I would not say that simply the complexity alone has something to do. I mean, I know where the question comes from, because, for example, if you eat very highly polished rice, or bread, white bread, which is very it's mostly starch. There is not a lot of complexity there, and there are not so many nutrients then there, right? So I get it for where the question comes from, but I probably would say that. Because we're eating anyway all the other foods, when I eat an apple or, I don't know, meat, I'm eating cells. So this is already highly complex as well. So I would say any type of not so processed food will provide enough nutrients. Yeah? I use a piece of microscope, okay. Hyphae, because there was a question, so not hyphas, hyphae, and hyphae are basically those long cells that essentially make up a fungus. And I don't know, if you eat mushrooms, for example, the mushrooms that we eat in food, these are actually only the fruiting body. These are only the parts that actually make the spores. But if you are, for example, out in nature and you see a couple of mushrooms growing, then actually the majority of the mushroom is in the ground in the form of microscopic hyphae, and collectively all of those hyphae are referred to as a mycelium. And you can see them, all of those long cells that you see over there, okay, must be part of this. So here as well. And they're kind of growing into the cheese and kind of fusing with the cheese, and therefore and the yeast that I showed you before, you could see that it's all a fungus, but it did not make those long cells. So you see there are different type of fungi around as well. Mycelium can span 300 miles or more. Yep, it can be huge. I was referring to the microbial ecology of the gut. I've read many people have poor microbial ecology. Perhaps kefir would help. Okay, I get it. As a matter of fact, it has been recommended. Well, not only kefir, but people eat yogurt because yogurt contains lactic acid bacteria. And I also wanted to add something, because you're talking about the microbial composition. This is some people who are on an antibiotic treatment. because they've got an infection, apparently this can also kill some of the microbes in the digestive system. And so apparently those people, to regenerate their digestive system, they're swallowing actually bacteria, freeze-dried bacteria, in capsules, to kind of regenerate the digestive system. Okay, so Hyphae. I don't know how do you. I need paper, and I don't have paper. I only have because the question is, how do you spell the Hyphae? So I'm going to use my this. So the Hyphae. These are the cells of a fungus. Okay. Yeah. So let me go back to the microscope again. Yeah. So and what we see here again is, we are actually, when we eat this cheese, we are eating mold, obviously. This is why some people consider cheese actually, strictly speaking, rotten milk. Some people told me. It's not quite wrong, and as a matter of fact, because the fungus and the fungi, they break down and the milk and to make cheese, this actually prevents other microorganisms from growing on the cheese. So this is actually a form of also preservation, because many of those fungi, they produce substances that prevents the growth of other, let's say, harmful microorganisms. Yeah. Everything we ingest except water is a living thing, or at least was once a living thing. Okay. For example, when we eat bread, then it is made of flour, which actually were seeds of wheat grains. It must have been a living thing, because it's an energy source, and ultimately all of the energy in a food chain comes from the sun, and at the very beginning of a food chain there are plants. What's your favorite organism to view under the microscope? My favorite organism, I have a favorite organism. It's I like to look a lot at ciliates. Okay, water organisms, because I like to look at them because you can actually look into the cell quite well, and you can see the events happening in the cell, and they're fairly large. Salt is not living, we eat it nevertheless, and of course I agree, and of course many other minerals as well. Okay, if we, for example, eat sugar, purified sugar and so on, I mean, certain soft drinks, Coca-Cola, it gives you energy, a lot of energy, because there's sugar in it, right? Water beers, of course. So let's move on a little bit. I want to show you now, and I would like to now show you the fungus here of the Camembert cheese. So what's going to happen is the following. I mean, this cheese was already cut open, but if you leave the cheese open now in a warm, not in the refrigerator, but in a warm place, you're going to see that the white fungus is going to start to grow over the cheese itself, okay, from the side. So this is and soon after a couple of days, sooner or later, this surface over here is also going to be completely white. And the cheese is going to become more and more soft. Why? Because the fungus will release its digestive enzymes into the cheese, making it soft, giving the cheese its typical cheesy taste. In other words, cheese is kind of pre-digested for us. Okay, what I'm going to do now is quite simply, I'm going to take a small sample of the white fluff from the surface and put it on the microscope slide, and let's have a look at that. So you already know how it works. I take a little bit of water. Here is some water. Here we go. And again, a toothpick. Again, you do not want to use too much. There's always the problem. If you use too much of this, then it's going to be too thick. It's going to cover itself, and you're not able to see the individual cells very well. So what I usually do is I try to take a little bit off here, scratch off a little bit of the surface, but sometimes it doesn't. It's a little bit a question of luck as well. Before that, I'm going to have to put on my glasses again. Here, this is much better. And yeah, let's see. Here it is. Okay, some white stuff. A little bit. Yeah, let's try to separate the cells a little bit as much as we can. Because if it's clustered together too much, then of course it's going to cover, the cells are going to cover each other, and it's difficult to see them separately. If it's a little bit clustered, it's fine. At least some of the cells should break off, and these are the ones that we can have a closer look at. Okay, so I hope that this is enough. And again, a cover glass. And again, I'm going to dry wipe it a little bit. Okay. Sometimes I'm also using not tissue paper, because tissue paper actually, there's some fibers left on here. Not a problem really, but there's also microfiber cloths used for cleaning the glasses. Yeah, if this happens, you don't see this quite well, but there is still, the cheese is too thick, and therefore the cover glass is not completely flat. So I'm just going to press down, okay, and press it flat. Okay, let's go for it again. Where is the microscope view? Okay, let's put the blue cheese away. And let's put the blue cheese away, and let's put the Camembert fungus into it. And let's have a look now. Where is it? It's too bright, way too bright. Here is the blob. So let's go up with the magnification, down a little bit with the intensity. Okay, close the condenser. Okay, 20 times now. Up with the light a little bit. This is the wrong setting. Here we go. That's too much, right, at the top. But look here on the bottom. Do you actually see again those long cells? Okay. So this is yeah, still too much. Actually, I had a much nicer one before, but you see over here it's better here on the side. Yeah. So let's go up 40 times. Too dark. Need to open the condenser a little bit. Here we go. I think that's much nicer. Okay. So you see the individual cells, those oval structures, maybe that these are also spores. Yeah? Or maybe they contain the spores, because you see those tiny circles. Yeah? But much nicer to be seen here is that those filamentous long cells that you see here, which make up the mycelium or the hyphae. Yeah? Yeah. So I'm going to, as we look at this here, I'm going to go up to 60. I'm going to again answer a few questions. So, and yeah, and here you see basically quite nicely the typical structure of a fungus. Those long filamentous cells, and I guess I would have to speculate. I guess that those round things in those cells here, that these are probably the spores. Okay, so let's have a look here. Again, a few questions. Have you ever tried taking a sample and leaving it in water at least overnight? That's how I find tardigrades in dry places. Okay, I think I'm too far down. Water bears. I've never found a tardigrade south of Europe, and it's so dry. Yeah. If you want to find tardigrades, maybe I'm going to make another live stream on just on tardigrades. You have to take moss, and you have to really leave it in a little bit of water. Don't flood it completely, because but leave it in water for a couple of days. Yeah. So then do you have an oil immersion lens? I'm asked. Yes, I do have an oil immersion lens, but I do not use it very often. Because once you are using oil immersion, I cannot switch back to the lower magnifications, because there's the danger then that the immersion oil will touch the other objectives. I don't like that. Yeah, so it's a little bit risky. Yeah, crystals. What about crystals? Yes, I've looked at crystals also before. Yeah, dried moss, not interesting. you gotta make the moss moist and wet, and this is when all of the microorganisms start to come alive again. Yeah, it was exciting to seeing the first water bear. Okay, yep, samples of moss, scrapings of tree trunks, rocks. Okay, yeah, yes, it will be recorded, and it will be made available later on as well. So it's 1 hour 14 already. So yeah, so this is basically now again in differential interference contrast in DIC. Let's have a look at those cells in phase contrast. Okay, let's just so that you see again how it looks like. So this is now DIC. Okay, and I'm going to change the filter settings a little bit so that you know what this looks like. Okay, and I'm going to, yeah, so that's, let's remember this a little bit here. And I'm going to find a place where the cells are more separate. Okay, and now I'm going to look at the same thing here using phase contrast. Okay, same magnification. Okay, I just have to change it over. So, and phase contrast, but I have to change the filter settings, and I have to change and open it, and I have to increase the light intensity, and maybe I'm gonna flip out the here, and this is, I need to refocus a little bit. This is how it looks in phase contrast, and you see, actually not so nice. The reason is because there is so much background, yeah, as well, yeah. But again, here you can see that the actual cells, they look darker than the background, but they have this white ring around them. Okay, so this yeah, allows you to see it. So now how does this look in bright field? You probably would like to know. Okay, so I'm going to switch over again to my bright field objective, and this is how it looks like in bright field. Yeah, you see that the contrast is, yeah, you can still see the cells, but the contrast is not quite as good. Yeah, yeah. Yeah, if you touch a piece of the adhesive tape to the fungi and then tap in the slide, you will get very, that's an interesting one. Thank you for the comment. What about trying to collect some? I know that you can use tape if you, for example, want to collect the spores of a mushroom, like the one that for cooking. Now I don't know if this is actually going to work well or not, but let's give it a try. Because why not? And look at this. I'm going to take some tape. I'm going to give it a try. Let's put it over the cheese. Ooh, yeah, sure enough, there is quite a bit here. I'm kind of concerned maybe it's going to be a little bit too much. I need to find a slide. But you know, you simply got to try it out. Okay, why not trying it out? And it looks kind of dense, but we'll see. Okay, so again, microscope view. I always change the slide when I go down with it. I always go down with the magnification first, so that I'm not crushing the objective into the slide. So it's too bright. Right. I'm switching over again to DIC, more light. It's very, very dense. But oh, look! Between, look at those lines here. That must be the hyphae of the fungus. Yeah, actually, this works quite nice. Thank you for the suggestion. See, 40 times. Let me go up with just a second. I need to always adjust the light intensity again. Ha, beautiful. Thank you for the suggestion. So look, haha, Camembert cheese. Camembert cheese sampled with a tape. This works better than my method. Yeah, look at this. Okay, so these are the cells of a fungus collected with some sticky tape. That's really nice. Okay, yeah, Thank you very much. This was in response to a comment. If you touch a piece of adhesive tape to the fungi and then tap that on the slide, you will get a very nice slide. Okay, thank you. Will you ever go back to regular condenser now that you have a dark field condenser? Well, actually, I have a DIC condenser. Yeah, I mean, I can go always back, and this is now in bright field. Yeah, so that's basically how most of you would be able to see it. Yeah, with DIC, I can play around with the color a little bit as well. See, I can give it all sorts of different colors. Yeah, but I rarely do that. Yeah, but that is now, this is bright field. Yeah, DIC requires a lot of light. Yeah. Face mites, yes, they're difficult specimen because face mites, I made a few videos already on face mites, but you have to have a little bit of luck on finding them. So there are not so many of them. Yeah, I love these streams. Please keep sharing your passion. Didn't get YouTube notification, by the way, got it late in heading. Yeah, but don't worry, because all of these videos will be made available later on. Yeah, so you can keep on watching them later. Yeah. Tried this trick with an ant and it was not good. Pity I don't have any good microscope or techniques such as DSC or phase. You don't need DSC or phase contrast all the time. Every technique has its own advantages and disadvantages, I would say. What you see right now, that is bright field. That's how you would actually be able to see it. And yeah, you see it, it looks perfect. I like the tape method. It's actually pretty nice. Yeah, and dark field, if you managed to do dark field, then the next thing for you to try out is Reinberg. Because in Reinberg illumination, instead of completely blocking out the central part of the filter, you just use a color, a dark color. You can use some nail polish maybe, or you can maybe print something on overhead foil and then use that as a filter. Yeah, and then you can actually not only get a white specimen on a black background, but you can play around with other colors as well. Yeah. Yeah. So yeah, no, I highly recommend take it a level further and try to make Reinberg filters. Yeah. DSC and dark field are expensive. Dark field not if you just make yourself a filter. And another thing that I think that's very important, because many of you are probably going to say, oh, DIC is so cool because you have you get this semi three-dimensional view and so on. Do the following: use dark field. A filter, and then swing out the filter a little bit so that the light only shines in from the side. You will get a three-dimensional effect. It looks very similar to DIC. It's called oblique illumination. The first thing that I would try out, get dark field and swing out the filter partially so that yeah, the light shines from one side and you will get an effect that looks very similar to DIC. Yeah? Yes, oblique illumination. Yeah. So this is yeah, something that I definitely would try out. Okay, maybe I'm gonna do another live stream on simply on different yeah, on dark field and Reinberg and DSC. Again, I made videos on this in my other channel, in this channel actually, in this very channel some years ago. Maybe you can search for it about filters, on making your own dark field filters and also Reinberg filters and oblique illumination filters. Yeah, so it's actually quite possible. Yeah. So this was actually a little bit longer than I intended. It is 1 hour and 20 minutes. Yeah, I just wanted to share with you a couple of microorganisms, fungi and bacteria that you can find on regular food. If you joined in late, a quick summary. I showed you the fungus of blue cheese, the fungus of a Camembert cheese, and yeah, yogurt bacteria over here. This here is yeast, a baker's yeast, which are in a dry form. And I'll also try to show you a little bit of yeah, the freeze-dried yogurt bacteria, but because they're in a dormant state, yeah, it takes some time for them to actually fill up with water and become active. So I think that the yogurt bacteria from fresh yogurt actually look much better than the freeze-dried ones, yeah, which take some time to become active. Yeah. So yeah, and the reason why I wanted to show you all of those microorganisms from food is of course, because we want to know what we eat, but at the same time, because they are safe, because I do not want to spoil food, or use, kind of, I don't know, bacteria from dead animals and so on. I don't know, dead insects. Of course, there's also decomposition going on, but that's not something that I'd like to touch, right? Or sewage water, wastewater. That's not stuff that I want to be in contact with, and therefore, especially when you do microscopy in an educational sector or at home for fun, so I recommend have a look at those things first. Yeah. So I would like to the last comments. How do I know if my yogurt has bacteria? If it doesn't say on the box, it will have bacteria in it, because that's what yogurt is all about. Okay. I don't know if it's possible to have yogurt without bacteria. Time flies when you have fun. 17 millimeters. Okay, central dark ring. Yes. Again, because many people are commenting about dark field, the diameter of the central ring in dark field, from my microscope, it was 17 millimeters, but generally what you can do is the higher the magnification, the larger the central ring should be. Okay, if it's too large, everything is going to be too dark. If it's too small, it's not going to be completely dark. So there is an optimum here. And I would say that there's a little bit of experimentation needed. In case of doubt, make it a little bit larger, because then the background will be dark. And if it's a little bit too small, then the background won't be completely dark. So you don't have this strong effect. So last remaining comments. Thank you for taking your time. It was an honor and a pleasure. I like doing these things. Educational as always. Yes, thank you very much. Look up fungi slide culture. You place a slide over the colony and culture it. The fungi will grow on the slide, and you can look at it. Only do it with safe fungi through at home. Yeah, that's an interesting one. Growing fungi on the slide. My 40 times dark field has a gray background and it looks like phase contrast. If it has a gray background, then this is because the central disk is not large enough, and because the dark field patch stops are made for maximum of usually for the 10 times objective. You have to make the central ring larger, then everything will become darker, however. So it depends on the numerical aperture and also on how high you put the condenser. So I wish you all the best. This was a little bit longer today, almost 1.5 hours. I liked it a lot, as always. I hope that you did as well. Do comment then later on as well when the video is then after this live stream. The video will be in YouTube to be watched and viewed, and then you can of course also post comments. Okay, everyone, all the best to you. Happy microbe hunting as always, and hopefully see you again next week. Bye bye.